Assalamualaikum warahmatullahi wabarakatuh. Our video is entitled The Next Generation Sequencing in COVID-19. What is Next Generation Sequencing? Next Generation Sequencing, NGS, is a modern tool for sequencing which is very variable and adjustable. What it is used for? NGS is used for studying disease-related regions in human and also tracking genetic changes in pathogenic viruses and bacteria. The outbreak of COVID-19 caused by SARS-CoV-2 virus has led to an urgent need of efficient and accurate method to analyze its genetic information. Conventional methods such as single sequencing is time-consuming and very expensive. NGS has emerged as a game changer when it enables fast and cost-effective sequencing of the virus's entire genome. This offers the crucial information of the evolution, transmission, and potential treatment target.
We move to methodology of next-generation sequencing of COVID-19. First, sample collection, clinical samples and isolate. Based on the article by Hurdle et al. 2020, there are two samples used for diagnostic testing of COVID-19, which are sputum, phlegm which coat up of mucus that collected from suspected COVID-19 patients. When arrived at the laboratory, the sputum samples were handled with biosafety practice by experienced lab workers using complete PPE, include FFP3 respirators and airborne pathogen-specific protocols. Second, viral DNA extraction. Total RNA was extracted using nucleomec kit on Kingfisher automate from clinical samples and using the nucleospin DX virus from isolates. Third, individual one-step RT-PCR for genome detection. SARS-CoV-2 real-time gene duplex reverse transcription RT-PCR was used to test sample of sputum and each isolate RNA extract. SARS-CoV-2 positive virus that cause COVID-19, if there are at least 2 out of 3 that virus gene targets were detected by RT-PCR assays. Then, for each sample, protoscript 2 first-strand cDNA synthesis kit was used to obtain single-strand cDNA from RNA extract. RT was done for 15 minutes at 48 degrees Celsius and 5 minutes at 80 degrees Celsius. Q5 hot start high fidelity DNA polymerase specific primer also used to produce amplicons from cDNA. After that, clean up of PCR products using AM-pure XP magnetic beads. The quality by electrophoresis in eGel EX 1% agarose and visualized with eGel PowerSnap electrophoresis.
Moving on to the fourth step in the NGS workflow, which is library preparation. According to Hurdle et al. 2020, one of the sequences that can be used is Illumina IC100 system. So what is library preparation? Library preparation involves fragmentation of DNA that undergo and repair to create blunt ends and are then ligated with sequencing adapters at both ends of the fragmented DNA. Later on, the non-bound adapter will be removed and the library was completed. After that, the sequencing library will undergo the PCR amplification. The sequencing libraries are loaded onto Illumina flow cell, which contains millions of small DNA fragments immobilized on its surface. The fragmented DNA are hybridized to complementary oligonucleotides on the flow cell, resulting in the formation of cluster. These instruments use a method called sequencing by synthesis. The sequencing occurs on a glass surface of the flow cell. Short pieces of the DNA called oligonucleotides are bound to the surface of the flow cell. These oligonucleotides match the adapter sequences of the library. First, the library gets denatured to form the single DNA strand. Then, this library gets added to the flow cell, which attaches to one of the two oligos. The strand then attached to the oligo is the forward strand. Next, the reverse strand gets made and the forward strand gets washed away. The library is now bound to the flow cell. If sequencing started now, the fluorescence signal will be too low for detection. So, each unique library fragment cluster generation from the library fragments need to get amplified to form clusters. This clonal amplification is by a PCR that happens at a single temperature. Annealing, extension, and melting occur by changing the flow cell solution. First, the strands binds to the second oligo on the flow cell to form a bridge. The strand gets copied, and then the double-stranded fragments get denatured. This copying and denaturing repeats over and over. Localized clusters get made, and finally the reverse strand get cut. This strand gets washed away, leaving the forward strand ready for sequencing. The sequencing of the forward strand involves sequencing primer binds to the forward strand. Next, fluorescent nucleotides GCTA get added to the flow cell along with the DNA polymerase. Each nucleotide has a different color of fluorescent tag and a terminator. So only one nucleotide that can get sequenced at one time. First, the complementary base binds to the sequence. Then, the camera reads and records the color of each cluster. Next, a new solution flows in and removes the terminators. The nucleotides and tDNA polymerase flowing again and another nucleotides get sequenced. This read cycle continues for the number of reads set on the sequencer. Once it's complete, these read sequences get washed away. Then the first index gets sequenced. The first index is read and washed away. If only a single read is needed, the sequencing ends here. But for the pair end sequencing, the second index is sequenced as well as the reverse strand of the library. There, the second index is read is no primer for the second index read. Instead, a bridge gets created so that the second oligo acts as a primer. The second index is then sequenced. These two index reads use unique dual indices. This allows use of up to 384 samples in the same flow cell. Next, the reverse strand gets made and the forward strand are cut and washed away. The reverse strand are then sequenced. Once the sequencing is complete, any bad reads get filtered out. The result will be analyzed and be read from bottom to top.
Next part is the pros and cons of next generation sequencing or NGS. First of all, the pros of NGS compared to the conventional method, which is the Sanger sequencing. NGS is high speed and low cost due to its functionality to sequence millions of fragments simultaneously in parallel at one time, which takes only about 4 hours to complete, while Sanger sequencing processes only single fragment at one time. Besides, analyzing the data in NGS is useful for clinical diagnosis and finding novel variant by expanding the number of target sequence in a single run. For example, discoveries of SARS-CoV-2 sequencing data allow the understanding of the transmissions of COVID-19, detecting the viral mutations such as Delta and Omicron variants that affect the vaccine potency, and identify this target strain for possible COVID-19 therapeutics, which are useful for the public health. The cons or limitations of NGS is increase the chances of contamination, which in this method it requires PCR amplifications of the template DNA, which may cause sequencing errors. This limitation can be overcome if sequencing is performed based on a single molecule without amplification. Lastly, NGS needs a high level of expertise because the interpretations of abundant data is quite challenging.
Lastly, in conclusion, next generation sequencing, which is the NGS, has been incredibly important in our fights against the COVID-19. This powerful genomic technology, such as the Sanger sequencing, has provided researchers with a comprehensive and detailed view of SARS-CoV-2 virus, enabling the rapid and accurate identification of virus strains. Next, NGS has facilitated the sequencing of the entire SARS-CoV-2 genome, allowing the scientists to analyze the virus genetic makeup, identify mutation, and monitor their spread. Moreover, NGS have been instrumental in development of diagnostic tests for the COVID-19. Last but not least, NGS have been accelerate the research effort towards the development of effective treatment and vaccine for the COVID-19. By studying the viral genome and identify the potential drug targets, researchers have been able to discover and develop of antiviral therapies.